Hi, my name is Rabiana Iqbal, and today I'll be talking about developing neurons for the class Psych 110 Brain and Behavior. Before I get into the content, here's a list of things I'll be talking about. These include the key terms, cell divisions, cell migrations, and then key takeaways from this video. Starting off, I'll be talking about the four key terms, which include neuronal proliferation, which is the creation of cells in the brain. There are two types, which are included in this slide. There's radioglia, which is one of the types. These are the first brain cells produced by asymmetrical division, and then there's neuron, which is the second type, which are nerve cells that allow communication between the brain and the body. Lastly, we have differentiation, which is the process where functions of cells become specialized. I'll be moving on to the two types of cell divisions. These include symmetrical and asymmetrical cell divisions. Symmetrical division results in two progenitor cells and the creation of a ventricular and subventricular zone. The ventricular zone will be important later on. Next, we have the asymmetrical division, which occurs 7 weeks after symmetrical division, and this results in one progenitor cell and one brain cell. The first few brain cells are called glial cells. This will also be important later on. Moving on to cell migration, there are also two types. The first one is radial migration. This occurs when brain cells follow the radial glia to the outer part of our brain to create the cortex. The cortex is created inside out, and the radial glial cells go through apoptosis, which is the death of cells, after providing guidance to the brain cells. So, the brain cells start off at the ventricular zone and they make their way up to the pia mater. And this image on the bottom right corner demonstrates what's going on. So the blue cells or the blue dots are like the first few cells, and they're at the bottom, indicating that it is the bottom inner layer is what is first created, and then slowly there are more layers adding up. And then, in contrast to that, we have the passive cell displacement, also known as a tangential migration. Here, brain cells are created outside in to form inner structures of the brain, such as the thalamus and the hypothalamus. This creation follows a spatiotemporal gradient, and this is also shown in the bottom right corner. The cells here in the upright triangles are represented as the first few cells, brain cells, and then slowly there are new cells added to the bottom. And then lastly, we have the downward facing triangles indicating the last few brain cells. So the outer layer is created first, and then slowly the inner layer is created last. So some of the key takeaways from this video include the creation of our brain isn't a one-step phenomenon. There are multiple steps, and my video only highlights the main ones. And it is a good first step to understanding how our brain is really created. Secondly, we have a brain that is still developing until our mid-20s. So it is crucial we engage in activities that improve our neural performance. Thank you for watching my video. Bye.